So, welcome back, Namrata and Shivani. Invite your old friends to join.
Okay, sir. Yes, sir.
So welcome back, students. So now number is 17 of you are here. So 6 are missing. So 6 of these are going to be, I consider that they are absent. And I will mark them absent at the last. Okay, so continue. I'm audible to all.
Yes, sir. Sir, I had a question. I was wondering um, if you were able to upload the slides
Yeah, I
on
didn't
the
upload it yet.
webinar.
I will upload. So don't bother that. I will take test only if you got the slide.
Okay, sir. Okay.
Okay.
Thank you.
So if I didn't upload the slide today or tomorrow, then simply means that I am not going to take test even on Monday. Okay.
Okay, sir.
So
Okay, sir.
don't worry
Thank you.
about uh, all the slides. Okay. So I will upload. So now coming to our module that we are talking about, and that is the cloning vector, and some of the feature of the cloning vector we talked about in that or the character they should have. Mostly the most important character is they should self-sustain, they should carry, and they should be recognizable also. Means you can find them. So this is a few properties that have. Then we discuss that plasmid can serve you as a vector. So the property they are having, so we make them to serve you as a good vector. So that's what is the reason we design or derive our vector based on the property of your plasmids. Then we talked about what the other property of the plasmids are, what this property make them, that is the size and the Fourier number. Uh, then we talked about the second type of the plasmid, conjugate compatibility and the conjugation. Also we talked about, and there are different type of plasmid. Then we mentioned and different features of the plasmids that we are going to be combined later. So when we develop a vector from your these plasmid. Then we talked about the bacteriophages, and as mentioned that they are simply bacteria viruses, and they are viruses of bacteria. Simply means they are going to infect the viruses and they replicate within the viruses, and when the replication is going to over, they come out. And the property that we are talking about the viruses is that structurally they have head and tail. Tail is a part which help in the attachment, and wherever a head is a part which contain the genetic material, which contain the DNA or RNA of your virus. Generally, in case of the bacterioviruses or bacteriophages, they are DNA in nature. And in case of human, we know that viruses are of both viral and DNA and RNA viruses are there, and many most important viruses are of RNA in nature. But prominent infectious virus of human are RNA, whereas in case of viruses they are of DNA. So this is a protein, filamentous protein. Then life cycle we talk about how this phage is going to replicate. Because this life cycle is the thing which make them useful for us. Because when they are how they are replicating, whether they are sustaining within the cell or not, and in what way we can use them. So lactic and the lysogenic cycle we made, we find that it infect and then leads to release of a new is having a new DNA molecule or RNA molecule. So in this property we can use it, and infection cycle is important to study how really it happens, because while you are developing a vector, we consider all these things, whether we want our cell to lyse or our cell to die, or we want a sustainable thing. So means your vector should sustain, your virus should sustain within the cell. So depending on that, we have to design we have to understand what different feature your virus is going to have. That means genetic at a genetic level. Then we move to the another type of the virus. So we discuss at this point, this level, that the lambda phage. So lambda phage is an example of the phage. So this lambda phage is a DNA virus, and it is infecting in the same way as we have discussed. It is all over having this that your lytic and the lysogenic life cycle. And so lysogenic, it is going to integrate. Then come to the next type of the bacteriophage, and the name of that bacteriophage is going to be M13. So the first thing for you that whenever you're using the term bacteriophage, this bacteriophage means we are referring the viruses of bacteria. Okay. Then if you talk about the independent virus, which virus you are referring, then for that we are giving the name. Like when we are referring lambda phage, that means we are talking about a virus whose name is the lambda phage. Now the next is the M13. So this M13 is another again a virus whose name is M13, and this is also of bacteria, and it also infects E. coli. So what property of it is that new phage continuously assemble and release cell means here the release of the viruses are there, but no lysis is there. It's not like that your lambda phage that it is causing lysis. So when the lambda phase completes its life cycle, it leads to the death of the cell. But in case of the M13, it do not cause the death of the cell. And one more important property is there, as in case of your plasmid, we talked about the plasmid extraction, means you need to take out plasmid from the bacteria. You need to employ that lytic cycle, alkaline lysis also. That for that you also ask some questions. But here it's not like that. Here it is automatically coming out of the cell. So means just so the while you are developing a harvest strategy, so you don't need a live cell. You need those virus particles which are secreted out. So from the supernatant also you can get the viruses. Okay. So this is the way how it is going to proceed. So like this M13 got infected, then M13 continuously shoot out from the cells. Now the M13 are coming out. Now they further infected new cells, and that it keep on replicating through the life. But the growth rate is going to be lesser in such case. Okay, so what are the when we take the vectors? So our vectors all are based upon what are these two types, the lambda phage and M13 phage. Both we are going to use to develop our vector. So now to understand what gene organization is, because this gene organization is the main core thing which make you whether what characteristics must be there in your vector. Because when we are saying vector means we can't keep extra luggage, extra load, which is not necessary, which is not required to us. So if some extra luggage is there, so because extra luggage is definitely going to be chargeable, like when you're going in the flight, so we don't want to give an extra charge. So for that we are removing our extra luggage. So extra luggage means what is the gene organization of this lambda DNA. So in this gene organization of this lambda DNA, we have to remove all the genes which do not require for our vector-like action. So vector-like action means invasion, replication, and repackaging. We don't bother the lysogeny, lytic, whatever. We don't bother. We only bother that is uh, replication and infection. So, as we all already mentioned, that what about this lambda phage? It is a typically head and tail phage virus. So, whatever the when we are referring bacterial phages, so most of the bacterial phages are having such type of structure, like one head is there, one tail is there, and the structure is polyhedral, and the size of DNA that it carry, the DNA molecule, that DNA molecule is going to serve for us as a vector is of 49 kilobase per inch size. Okay, and it is the virus which is very intensely studied, and that's why we are using in vector, many of our vectors as a vector for many of our technologies. Okay, so position and all genes lambda are known. So where what gene is located. So feature of genetic map of gene related function cluster in genome, that's already known. I think this all we have talked in. So these are the different cluster that are present and what different role of their genes are. Then come to the another form that lambda phage occur. It occurs in both the form, in the linear form uh, and the circular form. So in the linear form, it have a cohesive ends. So this linear form is present when this vector is present independently, means outside the cell, means inside your head, viral head. But once it enters, but it invade its host, it become circular in structure. So that and at a cohesive end we call this is of a 12 nucleotide stretch, and this cohesive end is we also call it the co-site or co-end. So this cohesive end is playing a critical role, as I mentioned, when we are talking about the packaging. So complement sequence strand often refer as sticky ends, and the cohesive ends base pairing between them sticks together. The two ends and DNA molecule become circular in shape. These cohesive ends are also called as a post-site. These play two distinct roles during the lambda phase infection. That I'm saying is the main most important role is that, that's why they are going to be an important factor or an important feature for serving them as a vector. The first thing is they allow linear molecules that inject into the cell to stay supplied. So this is a consequence is there. So there always a circularization is there. So in the bacteria, all DNA, whether it's going to be plasmid or it is going to be your chromosomal DNA, both are circular. So that's why it is in principle anything that is replicating in it must be circular. So that's why it also when enter the enter the bacterial cell, it also becomes circular in nature. So that is the role of this co-site. So when your DNA, this virus or this DNA enter the cell, this co-site help them to recircularize. And now second thing is which is necessary and prerequisite for on insertion. This, just a moment. Okay, so this is the first thing that we mentioned, and the second thing is the packaging. The second role of the foresight is rather different, and that is the that help in the packaging. So it come after the prophase has excised from the host genome. So now the replication is there, the lytic cycle is going to be there. So prophase is the phase when it is integrated. So that is of the lysogeny that we mentioned. Right? So when the lysogeny is over, now the phages are going to come out of the now it is going to play role in the packaging into the capsid. So at this stage, large amount of lambda DNA molecules are going to be produced. The replication mode is rolling circular because it is circular in nature, as your virus are going to replicate in. So continually rolled off and keep on replicating. So this result in the catene consists a series of linear lambda genomes joined together at both sides. So when they are replicating in a chain, you can say, and a rolling circular model means a circle is there, and that circle is keep on replicating and replicating and replicating. No replication is not stopping anywhere. So that replication is continuous. So that so circle have no ends. So then it keeps on replicating. It keeps on replicating. Okay, so here you can see in this image also. So how it replicate. So so it is a circle. So when it replicates, keep on replicating this action. When it reaches at this point, it again started here. When it reaches this point, then again started here. So in this way, a long stretch of DNA is being formed, and they are joined because if their ends are going to be cos ends, so they are joined at the cos sides. So now when this replication is going to be over, then endonuclease activity is required. So endonuclease help in the breaking down, cleaving it to the individual lambda genome. So here also the endonucleate talking about, they are also going to be specific type of endonuclease so that they cleave. So you can say that they are also a restriction endonuclease. So that's why they are cleaving at a specific site within your lambda genome. So when they cleave it, so you are having a double-stranded one. So then endonuclease activity leads to a sticky production, sticky ends. So as you are saying, the sticky ends are being produced. So it means the recognition site is going to be of around 12 base pair long for it, because this 12 base pair long sticky end is to be generated. So now what happened? Now this is as this both the end, again you are going to get positive ends are there, oocyte is there. Now this is an act of conjunction, and the other lambda protein start packaging each lambda genome into phage head structure. So wherever these co-sites are there, so whatever these co-sites are the recognition site. So this co-site is recognized by the proteins which are going to be part of your capsid. So they come to this co-site, they recognize, and both the end co-sites are there, and they start packaging around it. They start forming capsid around your DNA. Cleavage and packaging process recognize just the co-site and the DNA sequence to either side of them, changing the structure of internal regions of the uh, lambda genome. So whatever change you do in the inner structure of your lambda genome, if the co-site is intact, they will detect it and they will start packaging around the host sites. So simply the for example, by inserting a new gene has no effect on these events so long as overall length of genome is not altered too greatly. So overall length of genome we mentioned it is 49 uh, kilo base pair. So if the 49 kilo base pair is the length, so if you are not changing very drastically, like from 49 to you make to the 15 or 49 you change to the 80, so that's the drastic change is there. If slight change might be there, if you are removing some and inserting a new gene, then you can package it off. So the only requirement you need a co-site at each of the ends. So but that will be packaged. So in this way, when you're designing a vector based on the bacteriophages and more specifically this lambda phases, you must have a co-sites at both the ends. And other than that, that origin of replication is required here also. Now coming to the next one. So this is about what is about M13 phase. So M13 is not a capsid type of things. Here is a filamentous. So filamentous means the structure is the filamentous. So within this filament, you are going to have your DNA molecule, and DNA molecule here is a one great thing is that DNA molecule here is a single stranded. In previous case, we are talking referring the DNA molecule is double stranded. And all we know that generally DNA is double stranded and RNA is single stranded. But here in case of M13 phase or M13 bacteriophage, your DNA molecule, the genome of M13 phages is single stranded, and it is again one more thing is it is circular also. So it can help you in one more way if you require a single stranded DNA. If you don't need a double stranded DNA, if you need only one strand, then this M13 phase can help you. It's simply, you don't need to denature it, you don't need to separate those two strands. It automatically will provide you a single stranded DNA. Okay. So another property of it is they have a small genome than the lambda genome. So the lambda genome was 49. Here the genome size is just 6407 in length. This is a property of your M13 phase. So this M13 phase, how it is going to help you? So Smaller size means it have rooms for fewer genes than the lambda genome. So a very less number of genes are there. That's why because this is possible because M13 capsid is structured from multiple copies, just 3 proteins. You don't need a large number of proteins as we require in the case of bacteriophages. You just need 3 proteins. So those 3 proteins are the total component of your capsid. Whereas in case of lambda, it is 15 different. So M13 follows a simpler infection life cycle than the lambda. Here is no lysogeny, no lytic is there, so you don't need enzymes for that purpose also. So you don't, it is not integrated in the genome, so no gene is required for the insertion also. So requirement of this M13 phage is a bit less as compared to your phage genome. Then infection cycle, how this infection is going to be? Infection is more or less similar if you eliminate the insertion so or the lysis. So what happened? Once DNA, once inside the cell, single-stranded genome act as a template for synthesis of complementary strand. So the replication occurs when the single strand is there, a complementary strand will form. So now this complementary strand is going to be double-stranded. Now we have to become further a single-stranded one because when it have to package, it have to be a single-stranded form, which is going to be out. So as I mentioned, it is not inserted and it keep on replicating. So once it becomes double stranded, it keep on replicating and forming a double stranded copy only. So now within the bacterial cell, it is behaving like a plasmid, you can say, a double stranded circular DNA. Okay, so when the bacterium divides, with the division of bacteria, it also divides. So it is sustained and maintained through the generation of bacterial cell. But in case of lambda phage, daughter cells are not going to get this bacteriophage because they will undergo a lytic phase of life. So this is again the same feature. So around the new phase produced during each generation. So you know that life cycle of bacteria is E. coli is around 20 minutes. So in each 20 minutes, it is going to produce around 1,000 new phases. Now for the some more features of it. Attractive cloning, genome size less than 10 kb, so it's good. Within range of desirable potential vector. Double stranded replicative form. There, so it behaves just like a plasmid. Easy to be easily prepared and can be reintroduced by transfection. So the main thing is that it is within the stay within the plasma within the cell and just behave like a plasmid. Some other feature you can say that if you need a single stranded, I already mentioned you need single stranded, then it can be help you. Cloning in M13 is an easy way of obtaining the single stranded DNA. So therefore the hybridization, if you do this, if you design a probe, if you want to get a probe, you can get probe simply by the M13 bacteriophage also. Using fast display technique for identifying pairs of genes whose protein product interact with another. So this is what can help it. So this is about the viruses. These two viruses we have mentioned. It's not just these two viruses. Other than these viruses are also there. You can the cauliflower mosaic virus. And if you talk to even Dr. Sunny, who is working on the development of vectors only, so he is using apple scar virus. So any virus that you feel and you found that a property suits you for the designing of a vector and designing that vector can help you that can serve your job, you can design a vector accordingly. So most living organisms affected by virus. So there is very few species might be there for them. Viruses are not being reported, but most of species are there for which viruses are there. Whether it's a human, any animal cells, or a fungal cells is there. So in fungus also there are viruses are there infecting them. So you can design virus, you can find virus for that. So human adenovirus is a virus that is being used for a vaccine that DNA vaccine. So the vaccine that that one is your Moderna virus, and this also not the Pfizer and the Covishield that you are taking. They are the adenovirus based vaccine. So they contain the vector that you are talking. That vector is developed on the basis of human adenoviruses. And other viruses, the baculovirus. So many viruses are there. So whatever virus you found that infecting the host, you can choose that and you can use it as a vector. Condition, you have to remove unnecessary things which are like, if you found that this virus you are want to use this virus as a vector, but this virus is going to be able to cause disease. So in that case, you can't use that as a virus. So you need to remove those features which are factors which are behind the disease causation. Okay. Colimovirus, Gemini virus, these are the just Gemini virus. If you want to know about more to Gemini virus, you can contact to Dr. Sunny. He is working in the Gemini virus. Okay, so this is about the viruses and the cloning vectors that you can use. Okay, so now next thing that we have to talk is high capacity vectors cloning, that is YAG and BAC. So now I'm not continuing for this YAG and BAC because then it is going to be an overload for you. So just now I'm giving you time for the discussion. So any question that you feel that for the property of your bacteriophage and the plasmids that you need to know, and that is you didn't get a clear information, please let me know. I will explain them again.
So there are many strategies also. There are many experimental ones that are available which have explored the effect of it.
High capacity cloning vectors, will human artificial chromosome also come under that, or is it only yeast and
Sorry.
so for high capacity cloning vectors, is it only yeast artificial chromosome and back, or will human artificial chromosome also come with that?
Yes, so these we are going to talk about in next class. So this back and yak. So they are the high capacities. You can load a very large size of DNA over them. So they are going to be artificial chromosomes. So chromosomes, the property of the chromosome. So what do you mean by chromosome? If you talk about a human chromosome, whenever you say a chromosome, we derive an X shape. So this X shape structure is going to be a chromosome. So this X shape structure is going to have few features. One feature is that it has a centromere. Second feature is that it has telomere. Okay. So these centromere and the telomere, they are the required features. So other than the centromere and telomere, you need origin of replication, you need whatever the feature we talked about in case of the yeast and in case of the plasmid and the viruses, all should be there also. But a new feature in this case is that you need a centromere and you need two telomeres at both the ends. So if you have these centromere and telomeres, they are going to help you to arrange on the metaphasic plate, and when they got arranged on the metaphasic plate, then it to be pulled toward the poles. When they are pulled toward the poles, then in the next generation you are going to have a new progeny of, or simply a cloning of your DNA.
Okay, sir. Thank you.
Okay, so any other questions?
Whereas if they are not really
Okay, so if no question, then we move to our next part, that is uh, vectors for your plasmids. So what plasmid are vector, what property your plasmid vector should have or are having, and what different types of vector we are using. Okay, so just a moment, I'm sharing next presentation. Okay, so today I will discuss this in brief only. So because it's going to be a vast description is going to be required. So cloning vector now for the E. coli. So E. coli is our host strain which we are going to use for our cloning purposes. So now what type of the vector we should use? What are the different vector and how these vectors will be described? So the plasmid, as we mentioned, that they are the first and they are the simplest and they are widely used. Okay. So the property that one thing is that they should be small in size. So we are not using very large vector plasmid as a vector. We are using only small plasmid as a vector. So there are the large number of the plasmid vectors are there which are being used in case of E. coli, and you can get them from the market also. But main thing is that the feature that we consider that the plasmid vector should have. So these plasmid these properties are going to be there when we talk about vectors for your E. coli. That is high transformation efficiency. So what this transformation efficiency means? This transformation efficiency is the important thing that we will talk about in your practical also. So transformation means that your vector DNA is going to be outside of your cell. When you're giving whatever amount of your DNA, the vector amount of DNA you are having, even a small amount of DNA will be able to transform. Transform means it will be able to enter to the host. Transformation. That is called transformation. Okay, so one property that your vector should have is the transformation efficiency. Transformation efficiency simply means even if you are having a nanogram of your DNA, it must enter to the cell. One thing is that all the hosts are not compatible. All the hosts are not natural hosts of a plasmid. Like E. coli is not a natural host for plasmid. So all the E. coli are not the host for the plasmid. Okay, so when they are not the host for the plasmid, then they won't carry it. Now the thing is that our plasmid is having high transformation efficiency. It can sustain, it can maintain itself in a host which is not its natural host. So that is the property, desired property of your plasmid as a vector. Second thing is a convenient selectable marker. Selectable marker means you can select when you load your DNA, when you load your vector, your insert or a vector, you can select it when it is being transformed. When we are saying when to select, when it is present within the cell. So the property should be present for a vector that it can distinguish between ligated one, means in which your desired segment is being inserted, or in which segment is not inserted. And one more is that ability to clone reasonably large piece of your DNA. Means the product that you want to load over it, it should be quite large. Means up to 8 kb, because generally when we talk about the gene, our genes may range from a small gene around 1 kb to 8 kb, so depending on what size of your gene is going to be. So it must have some space so that it can accommodate that. Size is directly related, or you can say the size is inversely related with the transformation efficiency. So if your DNA is going to be, if your vector is vector plus product is going to be larger in size, transformation efficiency is going to be low. So it means you need a large amount of your DNA to transform, and even that chances are there, no transformation occur. Okay, so whatever the most routinely transformation or most routinely the cloning that we are going to refer, and for what we need the vector, we are relying on the plasmid based vector. Even we know that human, we are not having a plasmid in us, we are not a natural host of the plasmid, but still we are designing a plasmid that you can transform to animal cells also. So that is what property you should add to that. That we will talk about later when we are talking about the eukaryotic vector. But here simply you can say that these are the property your vector should have. Just hold a moment. Okay, so as I mentioned, so these plasmid vectors are going to be the routine vectors. So whenever you come to the lab, whenever you talk any of the lab, you will go, any of the work you will start of the molecular biology, you will start with a plasmid vector. So these are simply, you can say that these are routinely used vectors. So now, which vector is the first developed and how it was developed and what feature it is going to have. So it is the first vector that was developed is named PBR322. So the name this vector came, so how it is going to be came. So this P stands for the plasmid and B and R, they stand for the name of the researcher who discovered it, and the 322 is the number of this plasmid vector. So simply you can say that when these scientists were developing this plasmid, before their successful design, they developed 321 vectors, so which do not work successfully. However, it is the first successful vector, but it is not used nowadays because it lacks the whatever the feature. Because as we keep on advancing, we need an advanced technology. So this is of an old technology, old vector. It is lacking all the feature that we think, like your mobile phone should have camera also. It should have high memory also. It can do the 5G net, can work on the 5G network. Similarly, in case of plasmids are also there. So the back it is having all the property that you need, but it's not having many of the property also. Third property it is having, we'll talk in next slides. So it is nowadays we are not using it, and but its fundamental property that it is having, all the vectors that we are having till date, all the vectors are based on PBR 322 platform. Simply you can see certain slight change definitely may be there, but the platform that we use, the technology that we use, that is based on the PBR 322. So now, as I'm asking, I told you that this PBR 322, how this being named and how it's been labeled, so that the name PBR 322 confirm the standard rules of the vector nomenclature. P indicates the plasmid, B and R identify laboratory in which the vector was originally constructed. B and R stand for Bolivar and Rodriguez. So they were the two researchers who developed these vector. So you can simply say that BR is means the laboratory of Bolivar and Rodriguez. So that's why the plasmid Bolivar Rodriguez and 322 is the number. It's just to distinguish plasmid from other developed in the same lab. So this in the same lab, 321 plasmids were earlier developed before we reached to a successful plasmid, that is PBR 322. After this success of 322, They also developed few more plasmids, and they named that PBR 325, PBR 327, PBR 328. So these are the different plasmid they have developed, and the standard nomenclature, whatever the plasmid you are going to have, whatever the plasmid you are going to use, they are going to based on the similar nomenclature. Like P should be there, then two alphabet is going to be there. These two alphabet is going to correspond to the laboratory from which they are going to develop, then followed by the number or some other alphabet. Okay, so the property that it have. So the property I will think now we are at the end of our session. So I'm stopping here, and the detailed property of PBR 322 I will talk in next session on Monday. Okay, so till then stay safe, stay happy, and for the attendance, 18 students of you I have downloaded your list. So they are present, rest all are absent. So I'm just calling the name of those 17 students which were present at the initial phase. Just a moment. So please, those name I call, please turn on your camera. So Namrata, Namrita, 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 Shivani,
Present, sir.
Tridev,
Present, sir.
Manaswini,
Present, sir.
Tanvi,
Present, sir.
Vanshika,
Present, sir.
Vivek,
Present, sir.
Atoiba,
Present, sir.
Devanshi, Devanshi,
Present, sir.
Ayushi,
Present,
Ayush,
sir.
Nehal,
Present, sir.
Rupal, Rupal, Sahiba,
Present, sir.
Yuvraj, Yuvraj, Yuvraj,
Present, sir.
Shakti,
Present, sir.
Demetrius,
Present, sir.
Yukta,
Present, sir.
Sanjana, Sanjana, Sanjana,
Present, sir.
Sanjana, then you will raise the issue that you've been marked absent. I'm calling your name several times and you're not responding.
Present, sir.
Okay, so by the if you've been marked absent, next time also then don't blame me that I marked you absent. Okay, so the list that I called the names, so in it Rupal is absent, rest all are present. So these are the 16 students who are present today.
Hello.
Rest 8 are absent. Whose name I didn't call?
Sir,
Yeah,
you
Hello.
didn't call my name.
what's your name?
Adish. Adish.
Adish, at what time you joined?
Uh, around 2:20, I guess 2:23 around.
So what's the time of class?
Sir, 2:15, sir.
When?
Sir, I couldn't connect as it was it's still raining here.
It is
That's why.
happening daily.
No, sir, I
It is
usually be there. I usually
That's
am
what I'm
at the
saying. It is
time.
happening daily. I have to wait for you for at least 10 minutes to start the lecture.
So
the
usually
daily,
I'm on time.
usually you
Always
have
that.
the time, but usually it also happens that I have to wait for you all for at least 10 minutes, and some of you are joining with the even at the end of the session also.
Excuse me, sir.
Yes.
Sorry,
Yes,
sir, but sir, there was some issue with my MS Teams only. So I switched another device and then I downloaded the MS Team on my mother's phone.
ये
Sorry, sir.
नम्रता बोल रही है, इशिता बोल रही है?
Sir, Ishita.
Yes, इशिता.
Sir,
Sorry,
am
sir.
I audible?
So this is ये सब फिर सभी अपना अपना बहाना लगाएंगे. जानवी, what was problem with you? जानवी, आप लेट क्यों हुए भाई? जानवी?
Sir, am I audible to you?
Yes,
Yes,
sir.
you are audible to me.
Sir, I'm present. Actually, there was some glitch with my MS team, so I
ये
joined back.
IM का शक्ल दिखाओ ताकि पता लगे कौन है IM में.
नम्रता sir.
नम्रता आपका लगा दिया मैंने.
अच्छा, okay sir.
Yes, जानवी.
Sir, you
Yeah.
can take my name.
Yeah, at what time you joined?
Sir, I joined at 10:16. Sorry, 3:16.
3:16. So अभी तो 3 बज के
2:16,
7
sir.
मिनट.
2:16.
No. The
Yes,
list
sir.
that I am having, this list is the last join in this list is at 2:21. I downloaded this list at 2:22, and the last one joined at 2:21.38 second is Sanjana Sharma.
Sir, there was some network issues. I joined from the other device at that time. 2:41.
See, so once you join, it records the time at what time you joined. It is not showing that Janvi joined and left. It is showing that Rupal joined and left. Rupal is not there. So it's also showing that Rupal joined at 2:17 and left at 2:21. Don't think Rupal is here even now. Okay, so be on time. So today I am giving you excuse. So I won't give any excuse from next onward. So one is Kshitij is absent today, and second is Rupal. Okay, Kshitij and Rupal are absent. Rest all are present. So please turn on all of you turn on your camera while leaving the team. Whose name I didn't call? Just turn on your camera and leave the team. So thank you all. Stay safe. Stay happy. Good day. Hmm.